Alright, this is Dr. Hether for Daytona State College doing another experiment with the enzyme amylase and the substrate starch. The first one we're going to do is we're going to add starch to test tube number 1. We're going to add amylase and we're going to add a pH buffer of 7. And then what we're going to do is we're going to drop this down and we're going to freeze it. In the experiment before, we actually boiled the enzyme, and boiling an enzyme has the effect of denaturing it, causing it to be unable to convert starch into the simple sugars by changing the actual physical characteristics, the structure, if you will, of the enzyme. For test tube number 2, we're going to do exactly the same thing as test tube number 1, but we are not going to freeze it. In test tube number 3, we're going to add amylase, we're going to add glucose, which is sugar, and we're going to add a pH buffer of 7. Test tube number 4, we're going to add amylase. We're going to add something called cellulose. Cellulose is plant fiber. It actually makes up the cell walls of plants, and our body cannot digest it. Test tube number 5, we're going to add amylase, cellulose, and a deionized water. So we're actually not going to have a pH buffer, just neutral water. Number 3 is going to be pepsidase, starch, and a pH of 7. For this one, we have the wrong enzyme. We have an enzyme, but enzymes are substrate specific. They actually require their specific enzyme. Just because you have an enzyme doesn't mean it's the right one. So for test tube number 7, the last one, we're going to add cellulose. Actually, I'm not sure if I added cellulose. We're actually going to add cellulose and something called bacterial amylase. Bacteria can actually break down the cellulose found in plant walls, whereas we can't. So we're actually going to test that after we incubate it for 60 minutes. A good example of freezing would be if we take a raw egg and we put it in the freezer and then take it out of the freezer and put it on the counter and let it thaw. We can unthaw a raw egg and then cook it, but if we boil it, we can't unboil the egg. So raising the temperature changes it. It's still an egg, but the structure has now been different. It's been converted. But if we freeze something, we can actually give it time to thaw, and it will return back to its normal state. Now, if we were testing this right away after a frozen enzyme, freezing an enzyme has the effect of slowing down the effectiveness, so it's not going to be as fast. However, in this experiment, since we're waiting 60 minutes for the time to elapse, that's more than ample time for amylase to thaw and hopefully convert all of the starch into sugar. So we're going to be using the same test as before. Iodine, or IKI, is going to be the test for starch. Iodine in the presence of starch turns black. And we're going to be using Benedict's reagent. Benedict's reagent is a blue reagent, and in the presence of sugar, when you heat up Benedict's reagent turns orange. I gave the example in the last video that it actually is going to resemble Tang, a very, very bright orange. Test tube number 6, and the last one, number 7. So if we add iodine to the first one, like we did, number 1 is negative, number 2 is negative, number 3 is going to be negative, number 4 should be positive. We cannot break down cellulose. Number 5 is positive for starch, 6 is positive for starch, but 7 is negative for starch. 7 was the one that contained the bacterial amylase with the cellulose. Now when we add Benedict's reagent to each one of these test tubes and then heat it up, we'll see are there sugars in the test tube. And again, we have to heat up Benedict's reagent, but we don't want to boil the test tubes. At this point, it wouldn't matter because the starch should have already converted everything. And we're going to heat it up for just a second. Very good. So test tube number 1 is negative for starch, positive for sugar. Test tube number 2 is negative for starch, positive for sugar. Freezing the enzyme had no effect. It did not denature it, and everything was fine. It converted all of the starch into sugar. Number 3, we had amylase, glucose, and pH of 7. Well, we're going to be negative for starch because we didn't have starch. We actually had glucose, so we are positive for sugar. Number 4 was the amylase and cellulose. the starch remained. We cannot break it down. There's no sugar. The next one, amylase, cellulose. It didn't matter whether it was deionized water or a pH of 7. It still remained starch. Number 6, we added starch. We added pepsidase, which is for proteins. It's the wrong enzyme. And we're going to be positive for starch and negative for sugar. And the last one again showed how bacterial amylase was able to convert all of the cellulose into sugars, and this is the experiment.